This lecture will be by Dr. Fani Hatjina, and it speaks of the results of the European experiment on genotype-environment interaction. May I now tell you who Dr. Hatjina is? She works at the Institute in Greece. She is a biologist with a doctorate on pollination by the honeybee. She promotes the well-being of bees and is the director of the Animal Science Institute that are a part of DEMETER, a Greek agricultural organization, impact of pesticides on behavior and build of bees. Fani is a member of the International Bee Research Association Council, and that's a research association for over 10 years. She has coordinated the working group in APIMONDIA that deals with the noxious impact of pesticides on the honeybees, and she has been the president of the Commission for Bee Health in APIMONDI. She is not only a researcher but also a lecturer. She promotes epitourism and also environmentally friendly apiculture. The floor is yours.
Hello?
Słyszymy cię.
Hello? Can you hear me?
We can hear.
Tak, słyszymy.
Perfect. Hello, everybody. Dear organizers of this BEECOME conference and dear friends, I'm really very happy to be with you. It seems that BEECOME conferences become some of my habits. I have been participated in most of them, and I'm really very happy to be in this one as well. Thank you so much for the invitation. So I will continue quickly, not missing any time after so interesting things that we hear before, because I was trying to follow you. And today I was asked to talk about the results of this very big European project on environment and interactions with the genotypes of bees. But the title, as you can see, it's just a little bit different. But in fact, this is the reason why I'm with you here. And Anna, thank you very much for the introduction about to who I am, where I'm working. You can see a picture of the Institute of the Department of Apiculture with the olive trees around, characteristic of Greece. So let's go continually, quickly. Well, what are the facts? The facts are that the honeybee colony losses, they are increasing year by year, and they are questioning really our practices. The climate change also is showing its effect on our bees and on the production and the profit that fellow beekeepers have on their colonies. And usually what we hear from the beekeepers is that more and more they are dependent, we also see it through our collaboration, that they are dependent more and more on artificial feeding for their bees, veterinary medicines, but even also on queens from breeding centers, because many years ago, people, they were producing their own queens. Now many people, they are buying their queens, they get their queens from breeding centers. Also, what is very common nowadays is that we see new technologies coming in. New technologies are developed for the bees, in the beekeeping sector, but still, Varroa is the #1 enemy in terms of pests and diseases inside the colony, and we have to fight continuously with this pest. So the question is beekeeping, as we know it today, sustainable? Can be sustainable for a long time? And at the end, what is sustainable beekeeping? Well, if we think about the terminology and we take it by that, then sustainable beekeeping is the beekeeping of bees, is the keeping of bees in a way that we all respect the well-being of the bees in this world and their history as well. And together with this, the natural resources from where they can get their food for living. Also, at the same time, we need to produce high-quality products with less inputs in order to ensure adequate income for livelihoods, but not only for this, because beekeeping, it's not only because we want to produce products. Beekeeping exists because we need to have also adequate sources for pollination services. And this maybe is the greatest advantage of keeping bees around the world. We have a little bit of a delay on the slides, but yeah, there we are. So all around the world, beekeepers and scientists together, we all trying to find solutions and to explain why and what is behind the low losses sometimes, but many times behind the high losses of our colonies, or why there is variability that is experienced in the vitality of the honeybee colonies between the different regions. And the questions are, which are these factors? Is it only Varroa levels? There are other parasites that they are influencing things. It is also Varroa biology, how the different genotypes behave in different areas. So these questions, they are not new. They were in our minds some years ago, and then through Colos big association, the group that it was called Working Group for Honeybee Vitality and Diversity, now it's Bee Breeding Network. In 2009, we sit down, we discuss, we acknowledge the difficulties and all these questions, and then we develop a protocol that it was set up at, in a total of applied in 621 colonies around Europe in 21 locations. I'm sorry, something is, yeah. So the idea came from Ralph Buchler. He was the initiator of this from Germany, and partners. There were, as you can see, a lot of people that I would like to thank their participation in this. And altogether, we discussed the protocol. We published the protocol. We discussed the methods in details and how to apply and for how long. And this was a kind of research that was no funding behind it. We did it with our own resources, but the Colos Association at that time, it was a cost action, and it was financing our meetings. That was very valuable indeed, because it's different when you discuss face-to-face and different when you are on the other side of a computer, like today, for example. I wish I could be with you there and not behind my computer. So the test parameters that we decided to test, it was the queen survival, the colony survival, the number of occupied combs, so it's population of the colonies, the brood area, pollen storage, honey and food balance, the feeding and the harvesting, Varroa, Nosema, and viruses, because we were monitoring this as well. And then, of course, recognition of other diseases symptoms. In general, the behavior of the bees, like the hygienic behavior, but also aggressiveness and swarming. I must tell you that during this it was a very big experiment it did. And the first times that we did such a big task, we had no Varroa treatment. That means that what we were experiencing, whatever we were measuring, it was under this pressure that Varroa was there all the time. And what you can see on your left is that the genotypes that they were tested from different subspecies, for example, it was collected and exchanged from different areas, more than 6, I think, genotypes of Carnica, 2 of Ligustica, 3 of Apis mellifera macedonica, 3 of mellifera, and we also had Siciliana bees from Sicily. That was also very, very important. And we were all testing them in 21 locations in a way that they could experience for 2.5 years when we finished the experiment. They were experience all different types of climatic conditions, as well as management practices, because people tend to have different management practices, which is really very important as well. And you can see it on your screen. 621 colonies then as a total, from summer of 2009, when we established the colonies, till the spring of 2012. Still some colonies were alive at that period. I must tell you that the idea, the concept was that the local line, for example, in my case, you can see the back side of my institute here where we placed the colonies initially. In our case, the local bee was Apis mellifera macedonica, but then we had Apis mellifera macedonica from Bulgaria, we had Apis mellifera ligustica that came from Italy, and Apis mellifera carnica that came from Germany. That means that in each location we had the local population bred from our own colonies, and then we introduced 2 or 3 other lines, and they were all at the same location for these 2.5 years. If we had to move them, they were moved all together. Of course, before the experiment, where we started, and after the experiments, we tried to publish the results, not only because, okay, we are scientists, we like to have publications. This is our aim, but because we wanted beekeepers, to have the results as soon as possible, so you know what exactly we found. And all these articles are in Journal of Apicultural Science, which is the Polish journal, and Journal of Apicultural Research, and all of them, they are open access, so you can find them and you can read them. And some of them, they've been translated in local languages as well. So let's start with the first one. And the first question that we wanted to answer is how the bees survived. And from what you can see here, on your right, you can read that the local bees, the local colonies, they had a higher number of bees than the non-local ones, that's on average, but they also survived up to 83, I think, if I'm not mistaken, 83 days longer. That means that the survival of the local populations in each location, on average, was almost 3 months longer than the survival of the other populations, for different reasons that we will see further on. Then if we have a look in the next big topic, like the population dynamics and how they developed during the year, well, most of the populations, we are in Europe, we didn't have so many surprises. So most of the populations, they were going from the autumn, sometimes a little bit during the spring, sometimes a little bit down, but then very high up during the summer of the next year. And as I said before, the local bees always had, on average, a higher number of bees. What was very interesting also was that if you see, this line here represents the number of days that the average temperature was above 10%, because this shows how many days per year the bees were active. You know that above 10%, we put that as a threshold, and it showed that finally we could understand better the behavior of the bees in the colonies. And you can see that areas, we grouped the areas according to the weather conditions and according to especially this number of the days above 10 degrees, in 6 different locations, because in 6 different groups, because we had 21 locations. It's not easy to compare each other. So we wanted to see if they are in groups and they can show results better, to understand them better. So we group all the locations in 6 groups, in 6 clusters, weather clusters, as we call them. And you can see that in warmer areas, here is Greece and Avignon in France, and Unice in Croatia, that they are more Mediterranean. In general, the bees during and as an average for the all years that we measured, but also summer, spring, and winter, they tend to have a smaller population. So the colonies tend to be smaller compared with countries that it's more cold. And this is understandable, because you can see that bigger colonies, they can survive better the winter, because this is the most harsh period of the year. If we go a little bit more in details, and we can remember what I said before, that the local bees show better survival. If you remember, we said that on average, 83 days longer they survived, the local bees. But this is an example as a case study. I'm showing this here to you from Greece, that shows that Macedonica Greek, even compared to Macedonica from Bulgaria, that we are neighboring countries, we had about 70 something days difference. But compared to Carnica bees, it's almost 200 days higher survival for the local bees compared to the foreign genotype. And there is also a difference on the production of honey. This is, as I said, this is what it happened, what I experienced in our experiment here in our area. But of course, this could be different elsewhere. In general, though, it shows that the survival is higher from the local populations on the place of origin, and also their performance. It's better in honey production. And here you can see how quickly the foreign populations, they were diminished. We are still in Greece. We started in October, the first measurements in October of 2009. And even in October of 10, the Carnica bees had completely vanished from our experiment. We still had some Ligustica bees, only one the last year. And the Macedonica from Bulgaria, there were only 3, but they were a bit very weak, really. And the Greek bees survive better, but of course, because they know their place, and they don't know their place because their logic works like that, but because they are better adapted. And this is the aim. That was the aim of this experiment. But okay, we talked about survival, and we talked about the performance in honey production. What about Varroa infestation? What about the other parasites? And who are the reasons that we lose colonies or we lost colonies? As I said, we did not treat about Varroa, and you can see that in some of the areas, this is the Varroa infestation levels as an average of the whole years. You can see that some of them, they are really very high up, and this is in Croatia. In some areas, they were quite low. And then looking into the reasons why we had and what was significantly different between the locations and the genotypes, it seems that the location is one of the main reasons that differ between the performance of these colonies, and also, of course, our genotype, because shows in the model here, and maybe not other factors. So the number of adult bees also in the autumn, when we were measuring this one, was negatively correlated to Varroa infestation level in July, in August, and September of the same year. So, for example, you find that you have 2% of Varroa infestation level in August, you will know that this is negatively correlated for with the adult bees in September or October or November later on. That means that Varroa really makes a big damage on our colonies. And this way now we really showed that measuring Varroa during the summer, we can correlate it with the numbers of population or the population levels in following periods. That was in the same period, in the same year, but you can still see that Varroa infestation levels in October, of course, they are negatively correlated with the number of bees in spring 2010. So if you want to have strong colonies in spring, you need to have as minimum Varroa levels even in October. We don't probably need to wait until winter to kill Varroa or to fight for Varroa. Maybe we should think about keeping Varroa levels down even before October, before our colonies go into winter, into overwintering. And these results also helped us to understand that maybe we should change our strategy fighting Varroa. If we talk about defense and hygienic behavior, this experiment shows that the local bees were less aggressive and they were also showing higher hygienic behavior. I don't know if I need to talk about hygienic behavior, but just in 2 words for those that they are not very familiar with this terminology, it just shows how quickly the bees, the nursing bees, the cleaner bees can detect and remove let's say the sick brood or the dead brood from the colonies. So this way diminishing the pathogens inside the colony, sometimes also Varroa. And if you want to find all of these results together in a comprehensive way, you can find it in this article. It's in America Bee Journal, volume 155, number 6, in June of 2015. And you can see the summary of all results and you will understand better what I'm talking about. But I must tell you that it was such a difficult and huge experience. And for many of us, it was the first time to think how we can measure, how we can put down on a paper the performance tests and how we can evaluate the colonies, also for breeding purposes. So it was a vitality test, not only for the bees, also for the group. You can see it in a summer time or in a winter time. And in a summary, well, we have some delay here now. The interaction of the genotypes with the environment is very complex because what we did is to test different genotypes in different environments. And it's very complex indeed. There is no only one parameter that can influence it. But all these strains, the genotypes that we measure, that we evaluated, they showed strong signs of hybridization. So we could not say that we were 100% Ligustica or 100% Macedonica or 100% Carnica, but we had hybridization, which is something common to our knowledge in all Europe. But what is very important is that at the end, there is no genetic superiority of any bee population, but good adaptability. And every genotype can perform differently in different environments, as we saw. We observed also drastic differences in survival time and disease load, both between the locations and between the genetic strains. The local bees, as we said, survived on average 83 days longer than the foreign ones. That's on average. You saw that in the case study I showed you that individually you can find even higher differences. The most frequent evident, and for the reasons of colony losses, was Varroa, almost 40%. But then we also had problems with the queens because we lost the queens, they tried to supersede, and then they become drone layers, etc. But we also had nosema problems, especially in the south areas of Europe. Varroa infestations were influenced by the locations. We could not determine genetic origin on the frequency of the virus infestations, but for example, when my local colonies survived for more than 2 years and a half, well, they collapsed the last year afterwards, but still for 2 years and a half, they survived with high Varroa infestation level. Maybe that shows something that happens everywhere, that maybe they are better adapted to the strains of viruses. So even if they have Varroa, they can cope better with the viruses and the other pathogens as well. The local populations not only survive, but they also perform and fight disease better as one main conclusion. And our experiments demonstrated that the best bee that we are looking for, it's showing excellent performance and superior disease tolerance across all environments, does not really exist. You cannot take one population, take it everywhere, and it will perform the same way and best. So the best bee is the better adopted one, and maybe through breeding, we can make it even better. And these are the main results of this experiment. But this experiment also showed us and made us understand that finally how important are the local populations and what we can do to keep them, to preserve them. At the same time, we also try to bring down to your attention and to other scientists also methods and ways and protocols that we use to measure the quality of the queens and the performance test as well, and what is used in each country in each breeding program. This is another article also in open access in Journal of Apiculture Research in 2014. And this article, apart from the performance tests that we do in this level to see what are the best populations to carry on with the breeding or reproduction in the next years, we also looked into ways to evaluate the quality of the queen as individual. And you can see that you can do it through the way that they lay the eggs and what is the pattern of the brood and how many empty cells you have there, but also through anatomical characteristics that they show that the good queens that they produced with good food, but also with the proper age of the larvae, they have many ovarioles. Ovarioles are these tubes that they produce the eggs. And good queens, they have above one particular threshold, one number, above 130 per ovary, ovarioles per ovary. And of course, very, very, very important is the mating. And the mating of the queens shows how many, if there is a good mating, then it can ensure that in the spermatheca, which is this organ, the spermatheca, the queen carries quite a lot and also viable sperm from as many possible of drones, because this increases the vitality in the next generation. And one of the problems that we also face in Europe, especially in my country, that's why we thought different ways, is the density of the colonies, and that does not really allow to have control mating. And that is why, apart from instrumental insemination that people in Poland and my good colleagues and friends there, they do a great job. But we are not that familiar with insemination in my country. So we thought that maybe another way of having a kind of a control mating could help to pass on the desired characteristics. And I will try to share with you, this is the train of virgin queens, as we call it. I will try to share with you, I don't know if I can be able to do that. Well, I don't. It is a YouTube, oops. Anyway, I will do it after, so I will continue with the next slide now. Let's try again. No, I'm trying to upload a video. Well,
αρχίσαμε να εφαρμόζουμε ένα νέο σχετικά σύστημα φυσικής και συνάμα ελεγχόμενης σύζευξης των βασιλισσών. Σύμφωνα με το σύστημα αυτό, το απόγευμα στις 5:30 ανοίγουμε τις κυψέλες αναπαραγωγής κυφήνων, οι οποίες ήταν κλειστές με βασιλικό διάφραγμα από το προηγούμενο βράδυ, έτσι ώστε οι κυφήνες να μπορούν να πετάξουν ελεύθερα στις περιοχές σύζευξης.
So the selected drones are fly to the congregation areas for mating after the afternoon. We open them after 5 o'clock in the afternoon. We close during the day, and then at the same time we open the nucleus where we keep the virgin queens, and they are placed on these rails, so we can move them in and out of this cool very easily. Every afternoon for 1 week, 4 or 5 days, we do the same. We place them outside of the queens. You can see that they are on the lanes, on the rails, like the wagons of a train. That's why we call it train of virgin queens. This system is shifting the time of the queen mating some hours later of the time it normally happens, and at the time that there are no other drones available for mating apart from the selected drones. And the main reason we are using this, as I said, is that we have quite high density, not only in my country, it seems in other countries. And once the nucleus are dispersed in the field, the entrances are open, of course, and then it takes about half an hour for the swarm to warm up the queen to be ready to fly off. Just before dark, so they stay open about 3 hours. We close the entrances, we move them in the cool and dark box again, till the next day. And with special devices in front, we observe, for 2 years we were observing the flight of the queen, so we know what time they go out, what time they go in, how much time they spend outside, and how many times they fly for this. Inside the cool box, the temperature is 14 degrees to make the bees not move a lot, but not to be completely cold and form a cluster. And they are easy to move them inside. We avoid the artificial insemination, really very demanding. We still have some time.
Στη συνέχεια ξανακλίνονται με βασιλικό διάφραγμα και οι κυψέλες
And
αναπαράγονται.
again, of course, we close the drones. So initially that was called a Joe Horner system, but we name it the train of virgin queens. So I will move on,
Την κληρονομικότητα
if it allows me. Okay, so I will, if you allow me, I will finish this presentation just with an invitation for the next APIMONDIA conference, which is going to be in Ufa. It's the 47th APIMONDIA between 5 and 10 of September. And the system, it's already open for the abstract submission for my friends, colleagues, scientists, but also for and it's going to be focused on tree beekeeping, forest beekeeping. It's going to be really a bit than in other previous times. So join us also in APIMONDIA for 2022 because it was postponed. And it seems though in the forest or in very cold places or near the sea in islands, bees can survive good because they are good. They have a good adaptation. They have adaptation characteristics of the places where they come from. And that is the reason why we have so many different species around the world. And I would like to thank you very much for your attention, for your patience, and of course for your invitation. Thank you so much.
Thank you very much, Fani, for a very interesting lecture. We have a few questions indeed, and we will be asking them immediately.
Okay.
Okay, so first question. Are feral bees more...
Feral bees more?
Are feral bees more resistant to frost and diseases?
Very good question. We are really wondering and looking for the feral bees wherever they exist, because in many places there are not any feral bees. They should be. Feral bees or feral colonies, they should be more resistant to diseases. If they survive, that also means that they are more resistant to diseases, but also to cold and maybe extreme temperatures. Heat as well, because with climate change, the parameters that we get.
Thank you very much, Fani. Another question. Do we have any limitations in importing other populations of bees to Greece? Or can a beekeeper import any type or breed of honeybees that they think would be good?
Well, thank you. Thank you for this question. Because in Greece, beekeepers can import from any place from Europe. They could potentially import also from other places of Europe, but that's not easy because they also need to have a kind of a certificate that they are not Africanized bees and they are free of Tropilelaps, Tumida, and the small hive beetle as well. But from Europe, Greece does not control importations. Although we try to use their local species.
Thank you very much. Another question from the same person, I think. What was the year that the pollination started, and do beekeepers pay for it, and are there any incentives to use pollination?
Well, it is as beekeepers do not have any out of pollination contracts. There are no pollination contracts unless it's something that happens in a particular area. And I'm afraid that this happens because the density that inside of every crop the colonies. Still, there is more than 5 or 6 years now, the farmers have understood how important not only to have the colonies, the bee colonies, to have inside the orchards or at the edges of the contracts with the people, but not really. Unfortunately, they get paid only from the honey and that they collect. And I suppose if there are kind of under the law, that will bring us to the point of beekeepers respect more the use of the rules on the use of the pesticides, so we will have less problems.
Fani, czy możesz powtórzyć ostatnie zdanie?
Can you repeat the last sentence? I believe there is some problem with the connection and we couldn't hear you well.
Yes, I'm saying that there is also a delay. I'm saying that if, for example, in Europe, all around the world, these contracts are ruled and are set up by law, so this will respect more what we call protection of bees, and they will the rules on the use of the pesticides. So we will have problems. There are contracts, things that I also mentioned. I hope you have.
Tak, słyszeliśmy.
No?
Yes, we heard.
Więc może przejdę do
So perhaps I'll pass on to the following question.
Czy strefa klimatyczna wpływa
The
na
climate zone, does it influence in any way the way you fight varrosis? A very important question, isn't it?
Yes. It is a very important question. If think of we found that the climate zone cluster, as we define it, it is important on how easily or how Varroa infestations are growing, and also this determines the beekeeping practice and the methods, the frequency and the time of the year that beekeepers need to fight. In South countries, where we are more warm and we have a longer active season, Varroa infestation levels can tend to be very high up, and we need maybe more than 2 treatments for Varroa, at least what is happening now. Still, the idea of preparing and fighting Varroa before we prepare the colonies for the winter, it applies everywhere, no matter if there is climatic zone or weather conditions different. Because what we need to do is, even in the middle of the summer, we need to fight Varroa in order to have better production, higher production, and better health of the bees when they go to winter.
Bardzo dziękuję za wyczerpującą.
Thank you very much for such an extinguishing answer.
Bardzo dziękujemy za zapytania, za odpowiedzi.
Thank you very much for the questions, for the answers. Thank you very much for being with us and for presenting such a interesting lecture.